Today, we're going to talk about something that sounds like it's straight out of science fiction. We're going to explore the future of manufacturing, and it all starts with programming living cells. I want you to just imagine this for a second. What if our factories weren't giant buildings, but were actually microscopic? What if we could tell tiny living organisms to create everything from life-saving medicines to brand new sustainable materials? Well, that's not sci-fi anymore. It's happening right now. So here's how we're going to break it all down. First, we'll get into the big idea of programming cells. Then, we're taking a look inside a biofoundry. You'll see what I mean. We'll unpack the DBTL innovation cycle, that's the engine behind all this. We'll also see how scientists are teaming up worldwide. And finally, what this all means for the future. All right, let's start with the absolute basics. What does it even mean to program a living cell? Okay, it all boils down to this concept of a smart cell. And really, it's not as complicated as it sounds. We're talking about taking a super common microorganism, like yeast for bread or everyday bacteria, and then scientists go in and very precisely edit its genetic code, its DNA, to basically give it a new job. The whole point is to transform these little organisms into tiny living factories. Seriously, each individual cell becomes this miniature production line, churning out some kind of valuable substance we need. And the goal here is super practical. You're basically hijacking the cell's natural metabolism. Instead of just, living its life, you reprogram it to make high-value stuff, things like complex drugs or even health supplements. And it can do it way more efficiently. Okay, so if these smart cells are the little workers, where does all the designing and building happen? Well, that takes place in a really cool facility called a biofoundry. Now, when you hear lab, you might picture a scientist with a few glass beakers. I want you to throw that image right out the window. A biofoundry is like a biology lab that's been put on steroids. It's packed with robots, automated systems. It's all designed to do biological engineering at an industrial scale. You know what? Here's the best way to think about it. You know how a semiconductor foundry builds super complex computer chips? A biofoundry does something very similar, but instead of silicon, it's engineering life itself. It's a platform for rapidly designing, building, and testing new organisms. And the difference this automation makes is just insane. Get this. The Edinburgh Genome Foundry can handle over 2,000 DNA experiments in a single week. That is 20 times more than a person could do by hand. That kind of speed completely changes the game for innovation. So, what's the actual process that makes these biofoundries tick? Well, it's a special innovation loop that's really the heart of the whole operation. It's called the DBTL cycle, and it's a simple 4-step loop. First, you design the genetic tweaks you want to make. Next, you build the cell with those new genetics. Then you test it to see how well it works. And finally, you learn from the results, which feeds right back into the next design. You just keep running that cycle. Design, build, test, learn. Over and over, getting better each time. So what does this look like in the real world? Okay, let's take a concrete challenge. How about producing EPA? That's a popular health supplement, and omega-3. Right now, we mostly get it from fish, which isn't cheap or super sustainable. The mission then is to engineer a smart cell that can pump out EPA way more efficiently and for a lower cost. And what's so cool is how that simple 4-step idea becomes this super complex, data-heavy workflow. In the design phase, computers help figure out which genes to use. In the build phase, it's robots putting the DNA together. The test phase uses automated instruments to get the data. And here's the kicker. In the learn phase, machine learning analyzes all that data to suggest what to try next. It's this beautiful, self-improving loop. Now, this kind of technology it's too big for any one lab or even one country to figure out on its own. And that brings us to the bigger global picture. To make this all happen faster, leading labs from around the world created something called the Global Biofoundry Alliance, or GBA. You've got members from North America, Europe, Asia, Australia, all working together, sharing what they learn and trying to push the entire field forward as a team. And they're working together because they're all facing the same huge hurdles. I mean, running a biofoundry is crazy expensive. They need to agree on common standards so that software from one lab can work with hardware from another. And of course, you've got all the tricky legal stuff when you're sharing biological data across borders. The GBA exists to tackle these exact problems. All right, let's bring it all home. Let's talk about what this all adds up to. because the future this technology could build is pretty revolutionary. Probably the best way to grasp the potential here is to think about something we all know now, Moderna's mRNA vaccine platform, right? Traditional drug development takes forever and costs a fortune. But with a platform approach, which is what biofoundries are, you change the entire equation. Once you have the system down, you can pivot and design new solutions incredibly fast, whether it's for a new virus or a new material. And that just blows the doors wide open for potential breakthroughs. We're talking way beyond just new drugs. Imagine sustainable biofuels that don't rely on fossil fuels, eco-friendly chemicals, brand new materials. This tech could become a massive tool for tackling some of the world's biggest challenges, like the UN's Sustainable Development Goals. So, that really leaves us with the big final question. For the longest time, we've been learning to read genetic code. Now we're learning to write it. As we get better and better at designing biology, the question stops being, what can we create, and starts becoming, what should we create next?